Today is April 28th, 2014. My name is Miranda Byse, and I am in the Senior Physiologist Lounge in the San Diego Convention Center during the annual meeting of the American Physiological Society. Today, I have the pleasure and the honor of interviewing Dr. Richard Hawkins for the Society's Living History Project. Dr. Hawkins is a long-standing member of APS who focuses his research in physiological biochemistry. Richard, welcome to the Living History Project, and thank you for agreeing to be interviewed for this series.
Well, it's a pleasure to be here.
Fantastic. We are very happy to have you. So I was hoping that I would be able to ask you a few questions.
And surely.
Okay.
Surely.
So can you tell us about your family upbringing and how you became interested in science?
Well, yes. I was born in southern Connecticut just before World War II. And my father was an engineer, electrical engineer, who worked on, he was with the airplane industry. And of course, through the war, we moved around quite a bit. After the war, the airplane industry collapsed. But there was work out here in California, San Diego, so it's very nice to be back home again. And so he took a position with Convair and brought us all along with him. My father, being an engineer, was always interested in projects. So it was building things, wiring things up, telephones. And that, I suppose, initiated me into science of a sort. Now, I started, I thought I was going to have to learn a new language going from Connecticut to California, but that turned out not to be the case. So I went to school here, Cabrillo, which is about 2 or 3 miles from where we are now, and then to Point Loma High, junior high, they had junior highs then, and then Point Loma High School. And then I went to the only university in town, San Diego. It wasn't a university, it was a college, San Diego State College. It was a teacher's college. And it was a wonderful education. California really put a lot of effort into education. And so I got a very fine background in science. But I didn't know exactly, as many people do, I suppose, I didn't know exactly what I wanted to do. So when I finished in college, I was a good student, and I had a position then as instructor. Someone went on sabbatical, and so I became instructor of biology for a year. And I really had the good luck to meet Professor A. Baird Hastings, who had just retired as head of biochemistry at Harvard University. So he took me into the laboratory, and I became acquainted with biochemistry, and he encouraged me to apply to graduate school. But he says, it's too late. He said, look, you write to them and tell them that A. Baird told you to write, and drop in a transcript of your grades. Well, I was lucky enough they accepted me. So I went to Harvard started out in biochemistry in their medical science program. And that was fun. Biochemistry, I thought, was very interesting, but physiology was far more interesting to me. So I left biochemistry and went to, switched, and they were not too happy about that, but I went over to physiology because that was the science that put everything, or discipline, really, that put everything together. And Richard Berlin was my mentor. We worked on purine transport in leukocytes for the most part. And it took, I spent 4 more years there earning the degree. And I think the training, what was different, the training in, the education in California was absolutely superb technically. But the training at Harvard, there you learn how to put things together. They had people who were experts and who had, they had achieved, they achieved great things. And they taught the students, or tried to convey to the students, how to think along those lines. So I took the degree in 1969, and I still was interested in physiological biochemistry. So I wrote to Sir Hans Krebs, who in Oxford, and Sir Hans, and I asked him if he had a place. And I made it as easy as possible. I said I would do anything in his laboratory that he wanted me to do. And he accepted me if I would do anything that he asked. But at that point we had to get travel money from the national government, and you had to explain in a huge application why it was that you couldn't get the same training in the United States that you were trying to get in England. And I asked Sir Hans if he wouldn't give me a project. So he wrote it on one sentence. And I had to take that one sentence and build it up to 3 or 4 pages and convince the Public Health Service that the only place that I could really be trained was in Sir Hans's laboratory. And it all worked out. So I left there and joined his laboratory. I spent 2 wonderful years and became interested, not exactly in what he wanted to do, but I became interested in ketone bodies and the consumption of ketone bodies by the brain. So that began my interest in cerebral metabolism, and I've continued ever since. I wish that I had asked for 3 years, but I'd only asked for 2, and I was afraid. I said, if I asked for 3 years, they may not let me go. If I asked for 2, I was more certain. But then we had, I don't know if you know what a postal strike is, but in England, everything is run by the Postal Service. That means mail, telephones, everything. So they had a postal strike. So I was cut off from communication with the United States. I couldn't write, I couldn't call. And there I was with 1 wife and 2 children. And well, it was a distressing time in the sense I needed to get some place to work. And eventually the strike lifted, and it was Richard Veitch at the National Institutes of Mental Health who kindly made a place for me in his laboratory. And I moved from Oxford to Washington, spent 2 years with Richard Veitch, Dr. Veitch, who was an absolute genius. One of these very lively characters, had ideas and opinions about everything. But then, still with 1 wife and 2 children, I had to buy a house. I needed some money, so I moved to the Food and Drug Administration because they gave me a GS-14 on it instead of the 13. I had no prospects for anything better in the NIMH, so I left there. And I spent 2 years regulating medical devices, optical medical devices, which was important, but compliance and regulation was not what I wanted to do. There was nothing, it wasn't the creation of new knowledge, it was the applying of rules and regulation. So I spent 2 years with the Food and Drug Administration, and then on soft money went to New York University to departments of physiology and neurosurgery. And with Joseph Ransohoff and William Haas, we did some very good research there. It was very interesting on midbrain lesions and metabolism. But then, continuing the climb, I became associate professor at New York University. I spent 1.5 years there, but then an opportunity opened up in Hershey, Pennsylvania. And what had happened, the Hershey Foundation had donated a large amount of money, enough to build a medical school. And very clever people, they said, we'll give the medical school to the Penn State University, but we won't pay anything else toward it. So here it is, you have it, and you're going to have to run it yourselves. And so, but they had facilities, they had money, they had everything that a young scientist could want. So I went to Hershey, and the head of it was in the Department of Anesthesia and with physiology. And it was with the chair of anesthesia was a friend of mine from my Oxford days. We were postdoctoral fellows together, and he was interested in research. So we went, we packed up the family and went from New York to Pennsylvania. It was a big change for my wife. I was happy there. It's a wonderful place to raise children, but it's not New York. But the advantage was is they had every laboratory facility that I could imagine. And these were times when money could be spent on research and grants were available. That's perhaps the biggest change that's occurred since then. And I stayed there for, I guess it was 11 years. And one of the things two things we did, three things really. We developed this technique for measuring brain glucose metabolism using naturally labeled glucose. And I think that was the first quantitative description of glucose metabolism. And then using autoradiographs and computer techniques, we put the whole brain together. So once you had the whole brain together, you could slice it up any way you liked. And you could take 2 or 3 brains and add them together and do t-tests or analysis of variance on 1.5 million places, 1.5 million points, which statisticians are always telling you, you don't have enough points. So I said, well, here I have 1.5 million. Can you deal with this? I said, well, no, that's too many points. So the other thing is we worked a good deal on hepatic encephalopathy, and that's work that in the next few years in Chicago that I continued. But during that time, I took a sabbatical leave with Floyd Bloom here in San Diego. It was a very good time. Sabbaticals are a time for academic refreshment. So we really aren't necessarily, to go there with the idea of accomplishing something is one goal, but in Dr. Bloom's case, I didn't promise to do anything, but I promised not to cost him any money. And I wrote grants and several papers and enjoyed the kindness of Dr. Bloom in his laboratory, saw everything that they were doing. And then at the end of that time, or somewhere during that time, I came to a decision. The decision was, I've been at Hershey long enough. So when a position as chair opened up in Chicago, at the Chicago Medical School, which is not in Chicago, it's north of Chicago, about 35 miles. So I took it and became chair of physiology at the Chicago Medical School, and I really had a wonderful time there. They had promised me that all the teaching was good. Well, it wasn't good. It was dreadful. So I had nothing to do. Anything I did would improve it. Many of my colleagues there say, you can't recruit anybody here. And I said, why not? It's a beautiful new space. And the dean was cooperative, Marshall Falk. And I think I recruited 5 faculty in about 3 years. So we really turned it around. Chicago Medical School was a great place to be. And I'd become acquainted with the chief executive officer, Herman Finch. Now, he had been chief executive officer and chairman of the board for 32 years. He made it possible for that university to exist. So we were always arguing about one thing or another, and I think Herman's idea was to bring people closer. So there's a long search for a chief academic officer, and that's a position I took after 5 years as chair. And then I spent the next 10 years, I guess it was, in administration. But I never gave up the laboratory. I would work to, say, 3:30 or so, and then go up to the laboratory and continue my writing and my research. And I had a wonderful team put together who knew everything to do. So we'd design the experiments. These were, for the most part, animal experiments where you design them, decide what it is you wanted to do, and they would go on for weeks. And we get the results and then analyze them and write articles. And my son joined me as a PhD student, Paul. He was a genius with computers, but he saw no future for himself in that. So he decided that he wanted to work in the laboratory and take a degree, which he did, a very fine degree. Then in his case, he saw the light and went out and practiced medicine and became an anesthesiologist. So anyway, back to the administration was challenging in a way. It's sort of like, it turned out that almost every administrator at the Chicago Medical School was a member of the Department of Physiology. And if you think about it, we're interested in systems and how they work and how they're organized, and that's what we do. So it was a natural position for me. I was chief academic officer for about 5 years, and then Mr. Finch passed away. And true to fashion, he said, when asked if he had a succession plan, he said, it'll take care of itself. So, and it did. So I became provost for a little while until there was a national search for president. Then I became president. And now my children were graduated. They all had degrees. They didn't need me anymore. And I didn't have to be a bread earner. Well, I had to earn something. Still had a wife at home. So I gave up. I resigned in 2001. But then they couldn't find, presidents are a dime a dozen, but they couldn't find one. So it took them until 2003 to find another president, and I have given up administration. I do no administration whatsoever. It was an exciting time because I had the chance to pull together all the related health sciences into one spot, and to acquire the Scholl School of Podiatry, which is now part of our organization. They were downtown, they came up and they found a nice home with us. And subsequently the school has grown along in the related health sciences. At that point I went back to, well, I didn't go back. I kept the laboratory open always, and I kept it going, but getting research grants was harder. I shouldn't have stayed on the presidency as long as I did, because getting a research grant and doing administrative work was a little difficult. So, but during those years we managed to, we began some experiments on the blood-brain barrier, and we were looking for experiments that that didn't require the day-to-day activities. So we took the capillaries out of brain and separated them into the luminal and the blood-facing and the abluminal membranes. So now you could take each of those, and the membranes would roll up in little spheres, and at that point we could study the transport processes. And that had never been done, and it was so much work, no one was liable to do it. First you had to have an abattoir close by so you could get tissues of the brain. It took a month to get enough preparations to do a few experiments. So I had the field to myself. Actually, this is one of the lessons I learned from Professor Sir Hans Krebs. He said, if you have two areas, if other people are working there, they'll get the answers. We'll get the answers sooner or later. Choose the area where there are fewer people working. So I did. And we had many happy years with that until about 2006 or 2007, when all the money ran out. But I still had data to keep me going, and have been writing ever since. But getting money to do experimental studies for me is more difficult. And as we all know, there's no age discrimination in the United States. But as one of my friends told me, he says, I'm too old. They won't give me a grant. And he was younger than I am. So now at 74, I have ambition, but I don't have a grant. The other thing we did at the Chicago Medical School, which you asked me what was significant too, which is becoming more significant, is we pretty well described the brain. When the liver fails, the brain suffers. And we were able to prevent that from happening by some experiments. So now, what do I do now? Now I teach students respiratory physiology, cardiovascular physiology. I give them advice whenever they choose to have it. Whether they take it or not, it's up to them. And administration for me is over. Laboratory work, I hope is not, but we'll see. Or science is not. I do keep up a relationship with the University of Valencia, where I have many colleagues who are also from Krebs's lab. I have a position on the faculty there, although it's not paid, which is good. And so we keep up in that way. So my plans for the future are to continue doing what I'm doing now. I don't plan on changing. One of the wonderful things that Congress did several years ago is they removed age restrictions. So as long as I can as long as I can fog a mirror, in essence, as long as I can keep going and giving lecture, I'm theirs. And so I have a position. Now, I think the only reasons that that the administration would be able to, or sometimes the university would they'd like to make changes. Of course, they always do. I understand that. I've been in a position where I had to make changes. But unless I'm going to commit moral turpitude, which sounds like a great deal of fun, but I'm not about to do it, unless I'm incompetent, not yet, and unless I abandon my duties, which I'm not about to. I live a mile away, so I can be from my kitchen into my office in 5 minutes. I'm going to continue doing what I'm doing. And I want to thank you for this wonderful opportunity to say a few words, to recapitulate my career. I'm sure I left a lot of it out, but I think that's probably a good thing.
So one thing is, in your career, you had a variety of positions and you did a variety of things. So what kind of advice would you give to students as far as, how they can succeed in physiology and how they can succeed in multiple career paths?
Well, the advice I give, or if I were to do it all over again, well, actually I did. I always had goals, and I knew what I was going to do. Establish long-term goals and milestones to achieve them. If you don't, you'll never achieve anything. Administration, I don't advise, except I was lucky because I had a team. I had technicians that went with me from Hershey to Chicago. I had people who stayed with me, and really they were the individuals who made it all happen. We get together and they're co-authors on almost all of my articles. So, but it's I can't recommend doing administration and laboratory work. It's difficult. It wasn't difficult at a particular time when there was money to do it. Now it's a full-time job to get the research dollars to adequately. But then there's other things that happen. The restrictions for animal use are so severe. When I was a student, a rat cost $1.25. We wanted them, we buy a dozen. And we go down anytime we like and get them. Well, now it's $50 plus transportation, and it's very expensive to keep them every day. So those types of experiments, which is what I enjoy doing, are very restrictive. So very difficult. But the other thing I tell young students, I tell them all the time, I said, you should go into physiology because you can work in pharmacology, whatever, but take your degree from physiology because we know what that is. If you come to me and tell me you're a physiologist, I know you know something about the cardiovascular system, you know about endocrines, you know about the respiratory system. If you tell me you're a neuroscientist, and I'm a member of the Society for Neuroscience, I don't know what you know. You could be a psychologist, you could be very narrowly trained, and you're not going to get a position in academia as easily. But the other is, don't overlook the other fields. I've had several students now that went into high school teaching, and physiologists are for the most part comfortable with mathematics, with physics, and with biology. So they fit right in. They are a desirable quantity. I've had some that have gone into writing, and They're skilled at writing scientifically based articles, and some who've gone into the pharmaceutical industry. But the first rule of science, of laboratory science, is not to fool yourself. Follow the data. So do that and decide your long-term goal, and you'll do well. Anyway, I've had a very long and happy career. I have no regrets whatsoever.
That is fantastic to hear, and that's excellent advice. Thank you. One other question is, what role has APS played in your career?
APS has been a guiding star, really. Before you were born, we'd go to the APS meetings in Atlantic City. In Atlantic City, probably nobody knows of these. There was a long boardwalk with the steel pier and lots of very decrepit old hotels. And there were seminars or sessions going on all the time. And in order to have your abstract accepted, you'd send it in and they would review it. So you'd send, 10 people may send abstracts in and only a few of them would be accepted. So if you're accepted, it was quite an honor. So they started me out early. My first presentations were with the APS. And then I think, it's always been my inspiration. I've stayed in contact with them and my colleagues with the APS. Later, I had the opportunity to serve on the Central Nervous System Committee and on the Long Range Planning Committee, and later on, when I retired the presidency, on the Finance Committee. And those were all good things to do, but it was always the APS that was there to continually providing the opportunities. They sponsor many of the symposia, and that was another opportunity that I took advantage of. We had a symposium in New Orleans on the blood-brain barrier, and the APS has been incredibly successful in keeping the science going. The other, you don't think of these, but financially, the American Physiological Society is incredibly well run, and it's stable. So where the other societies go up and down, and I'm not criticizing them at all, I think they're wonderful, but the APS has been sort of the beacon, the father of all the societies. And besides, remember, they give the Nobel Prize for physiology or medicine.
It's very true.
Anything else you'd like to know?
So what kind of changes have you seen in science or in the society during your career?
Well, it's hard to recognize the science as it used to be. Probably I've seen the biggest changes happen to be with the government regulations and with the restrictions that we have. Every time, there's one thing I learned with the Food and Drug Administration. You're completely isolated, and you say, well, they could fill out this form, or they really should be doing this too. Well, you don't realize as a bureaucrat how much work that's going to cause to those who are doing the work. And that's become onerous. There's almost, and many of it's well-intentioned and many of it's good. Of course, the chemicals should be safe and they should be stored safely. Radiation should be taken care of. Animals should be well cared for. All of these things, but the restrictions that come across us have been rather onerous. So that's one of the changes. The other change, when I was a graduate student, calculations were done on a slide rule and an abacus. There were no calculators at all. The first calculator, or the first small computer I saw, was when I went to Oxford. And they had an Olivetti which had 128 programmable steps and 4 memories. But you became very clever at writing your programs. But before that, calculating something with counting, scintillation counting, getting DPM, or separating dual label experiments, impossible. Well, not impossible, it's just that I used to disappear all day Saturday and all day Sunday to make calculations which now, you could get on the tape and read out. So probably it's the technical revolution, and of course we moved on to that to exchange of data with through the electronic sources. That's all changed very much. I remember asking Professor Aberd Hastings, I said, how many drafts of an article did you do? He said, I type it out once, and then the secretary would type it, and then I would go over it once, and the second draft was the one that was submitted. That's it. Now with word processors, we go over and over and over and over again. The other thing which is really many things have facilitated science. One is access to references, which began about 1965, 66, maybe a little bit later, where you can find references. I mean, you can, and through PubMed, you can get all of the references. And later, I don't know who championed this, but you can get the articles. Now that's a problem too if your journal is supporting the organization, such as APS. But on the other hand, from scientists, if you can get the article and read it right then and there, that's a wonderful advance. We like that. But how to pay for it all is a major issue.
Yes.
What else would you like to know?
I'm just going to ask, this is sort of a variation of a question I asked again, but I want to get it, so I have it. What advice would you give to students starting out in science today?
I would probably tell them this. First we would have a discussion, and it wouldn't be somebody dropping in the office and saying what advice I would give, because I would ask them to come back or spend a good time with them. Ask them to define their goals and then come back again and what they really wanted to do. Because whatever you choose to do, you have to be able to open your eyes and look forward with enthusiasm to that particular subject. So, it's a hard decision. We make decisions early in our life that are going to last a long, long time. So it's important to think as much about it as possible in the beginning. And then I think, look, science for me has been a wonderful career. I've enjoyed it. But you also should realize that the goal, or many of the goals in science, is to participate in science. Sooner or later in biology, everything will be discovered. But it's the participation in that discovery in any of the roles that you'd like that make the difference. So, and that's, look, many of the things that I've done or discovered, somebody else would have. It just maybe not exactly the same time. But I enjoyed doing it, and I enjoyed putting it together. And when you've discovered something new that you've never seen before and that no one else has seen before, that's the excitement of it. So, and you get a buzz out of it, but then you move on to the next thing. But I don't know if they exactly, it takes time. It would be more of a discussion to find out what that individual wanted to do, what his interests were, what his skills were. So I mean, it's not one piece of advice that would fit all individuals.
Okay.
Did I answer the question?
Yes, you did. Thank
Good.
you.
All right.
And then I'm just going to do the final spiel. So Dr. Hawkins, thank you very much for being with us today. It's been a pleasure speaking with you, and thank you for taking part in the Living History of Physiology.
Thank you, Miranda. It's been a real pleasure for me to be here, and the opportunity to express my gratitude to the American Physiological Society for all that it has done to foster my career.